All right, team. Here we go again. We are back in the throes of Mendelian genetics. And we will likely get through in the next 50-minute lecture. We'll look at two other kinds of problems. And then there'll be one more lecture, which will be me working through the one of the worksheets. And I'm going to show you where those are while I'm thinking about it. So once you've watched this video, you should be able to work these sheets in here. And if we go down to, let's see, where is it? It's in this next module. So in module 5, there's the genetics PowerPoint. But then there's also something that says more genetics problems, still even more genetics problems. So there's 3 sheets in here. And once we're done with today, I think you should be able to work any of these problems. So click on the sheet, open it up. You don't have to print it, but you can just open it. And I would recommend just doing the problems on a scratch sheet of paper. And then I will do some videos, at least with 1 or maybe 2 of the sheets showing you how the problems go, but it would be good if you worked on the problems before you looked at the cheat video, okay? All right, team. Let me just turn my clock on here so I know how long we've been going. It's always something, isn't it? Okay, so we're going to do a dihybrid cross now. Okay, we talked about sex determination last time and how, because of the nature of sex determination in us, the sperm, not the egg, is really what controls the sex of the child. Ironic, you know, there was that king that lopped his wife's heads off because they couldn't bear a male heir. Well, guess what? It was up to the king to donate that wise sperm, right? So shame on him for doing that. Okay, now here's the thing. Now we have something called a dihybrid cross, and it's a little bit more complicated because we're going to look now at 2 traits instead of just 1 trait. And we're going to follow those 2 traits from parents through the offspring like we did before. Okay, so let me just tell you, fundamentally, the way you work the problem and set it up is not any different. You're going to still go through all the same steps. You're going to assign alleles. You're going to determine the parent genotypes. You're going to determine the gametes. You're going to fuse those gametes together. So you're going to do all that same stuff. It's just now you're dealing with 2 traits instead of 1. And let me explain what I mean by that. So this problem says in squirrels, fluffy tails are dominant over scraggly tails. And the fun thing about genetics is you can just make stuff up. I don't know anything about squirrel genetics. I'm just making this up, okay? Just for fun. So we have fluffy tails are dominant over scraggly tails. And so there's a second trait. Red color is dominant over albino color. There are actually melanistic squirrels, albino-looking squirrels running around. I've seen them in Brevard. And now it says cross a homozygous dominant squirrel with a homozygous recessive squirrel and then determine the phenotypic and genotypic ratios of the F1 and F2. If there's any word in this problem that you don't know, then you need to go back and look at the vocabulary. All these words at this point in the game you need to know. Okay. So what we're dealing with are 2 traits here, and so we still have to assign alleles. And so fluffy is dominant, so we'll use a big F for fluffy and a little f for scraggly. And then red is also dominant, so we'll use a big R for red and a little r for albino. So we've assigned the alleles. Now, there's two different ways that you can actually think about this, okay? And I want to explain both of the ways. One of the ways you could think of it, and this is a possibility, depending on the traits, it could be that both of these traits are on the same pair of chromosomes, because chromosomes have lots of genes on them, right? And so it could just be coincidental that the tail trait and the color trait happen to be on the same chromosome. And we could say, here's the color trait and here's the tail trait. When this happens, we say they're linked. And there are lots of linked genes, right? Some genes are linked with others. The implications of that are, and we could just say maybe this parent has a big R and a little r there. Oops, yeah, that's the color, okay. And then maybe it's two little f's, so it has a scraggly tail, just as an example, okay. Here's the thing with linked genes, where one gene goes, the other goes. They're stuck together, right? So that is, in fact, one way you could think about this problem, but that is not the way that I want you to think about it. I don't want you to think about it like these genes are linked together, okay? Instead, I want you to think about it like this, that there's one pair of chromosomes that has the color gene, and then some other pair of chromosomes somewhere down the line has the tail gene. And so what you then realize is now you're dealing with 4 chromosomes for each parent instead of just 2 like we had before when we did a monohybrid cross. Other than that, you're going to work the problem just the same way. Okay, so now let's see if we can, from these words, figure out the parents' genotypes that we're going to cross together. Okay, and just be aware that they're not linked. They're like this. There's 2 chromosomes controlling color and 2 controlling the tail trait for each parent. Okay, so it tells you in the problem, cross a homozygous dominant squirrel, and you're going to now assume that it's homozygous dominant for both the traits with a homozygous recessive squirrel. So the traits, because they're not linked together, they don't have anything to do with each other. You can forget about one of the traits while you're dealing with the other. Okay. Looks like my alleles got erased. It's okay. There we go. Okay. Let's take the homozygous dominant squirrel. If it's homozygous dominant for the tail trait, what's its genotype? Well, homozygous means 2 of the same, dominant means they're uppercase. So we're going to cross a big F, right? And then it's homozygous dominant for the other trait, and red is dominant. So we're going to cross a big F, big R, with a homozygous recessive squirrel. So homozygous recessive for both traits, little f, little r, little r. So there's our parents, right? We determined our parents. Sometimes the most difficult, challenging thing you can do in one of these problems is figure out the parents, right? And we were able to do that here. So what do the parents do? They make gametes, right? So we're going to go, in a second, we'll go to the next slide. So that's where we're starting with those parents. So assign alleles, we did that. Here's the homozygous dominant parent. Here's the homozygous recessive parent. We did that. Now we cross them together. Now here's the thing. Now we have to figure out gametes, right? And what you have to realize is these letters represent 2 separate pairs of homologous chromosomes, right? So when meiosis occurs, remember how these things line up opposite one another and they're going to get pulled apart. One of the F's is going in one cell, one's going in another. Same thing with the R's. So forget about the R's for a second and tell yourself, what kind of gamete can we make from the F's? Well, they're both big F's. So the only gamete you can make is one with a big F. And then you forget about the F's and you go to the R's and you say, okay, when these R's pull apart, what are we going to get? Well, there again, the only thing there is a big R. So the only gamete that this parent can produce is a gamete that has a big F and a big R. Okay. Does it make sense that the number of chromosomes in the gamete is half that of the parent? It certainly does, because we did meiosis, right? Meiosis reduces the number of chromosomes. That's how we make the gametes. Okay, so same thing here, except little f and little r. Now, granted, I accept the fact it could be this little f and this little r, or this little f and this little r, or this little f and this little r, or this little f and this. It doesn't matter. Don't torture yourself with all of the repetition of the same thing. Okay, so now what do gametes do? They fuse, right? And put the F's and the R's back together. It's called restoring homology. You put the letters, the same letters back together. So don't write big F, big R, little f, little r. Put the big F and the big F and the little f together. Put the big R and the little r together. Now we have our F1 generation. And this represents a whole generation of squirrels. I don't know how many little baby squirrels mama squirrel has typically. I would imagine 5 or 6, okay? They're all going to have the same genotype with respect to these two traits, right? What is their phenotype? They're all the same, and what do they look like? What about for the tail trait? Fluffy, right? Because fluffy is dominant. And what about for the color trait? Red. So all the little baby squirrels are going to be fluffy and red. Okay. Now this problem asks you to go to the F2 generation. So we got to go to the next generation. Remember how we did that? We do a self-cross. Sorry. Of the F1, and now we're mating a little brother and a little sister together. I know that's weird, but that's what we're doing. We're crossing a girl squirrel and a boy squirrel from this generation together to get to the F2. That will take us to the F2 generation. It's just on paper, y'all. We're not doing it in real life, okay? I need to learn more about squirrels. I don't particularly like them. They're kind of annoying to me. Okay, so how do we get the F2 generation? Now, here's the part where students often get stuck. And I don't want you to get stuck, okay? So here's the deal. Here's one of the parents. We can say, okay, here's the boy squirrel, and here's the girl squirrel. They just happen to have the same genotype, right? Now, at this point, we have to do meiosis again to figure out the gametes, right? Now, remember, it gets one of the F's and one of the R's, but now we have choices, right? There are choices because there are a big F and a little f and a big R and a little r. So check it out. Think of all the possible combinations here of the F's and the R's. Okay, so one possibility is it could get a big F and a big R. It could get a big F and a little r. There's an equal chance it might get a little f and a big R, or a little f and a little r. There are 4 possible gametes here that this parent can make. So look at it and tell yourself, do not torture yourself by writing those 4 and then writing these 4 as well. Don't switch the letters around and think it's 4 more. That doesn't work that way. No, don't do it. It doesn't matter what order you put the letters in, okay? So be aware of that. Don't make extra work. You're going to get the wrong answer if you do that, okay? So what about this parent? Well, same thing, right? It's safe to say if the 2 parents have the same genotype, they make the same gametes. That's just the rule. Now, you may wonder, is there a way to know how many they're going to make? And there is actually a formula, and I'm going to give you the formula, and you might find it useful. The formula is 2 to the n This tells you the number of gametes that you're going to get from a parent. And it's to the n where n is equal to the number of heterozygous alleles. Okay, what am I saying? Well, remember, a heterozygous allele is one of each, right? So how many heterozygous alleles does this parent have? It has one set there and two, so it has one set of heterozygous alleles and another set. 2 squared is equal to 4, and there's your 4 gametes. See? So now, you know, is that helpful? Yeah, it might be helpful. At least you know how many there's going to be. And so if you start trying to figure them out, you know what your target is. But imagine if we did something like this. Okay. And I said, okay, now we're following 1, 2, 3, 4, 5, 6, 7, 8 traits. Oh my goodness. Across, you know, parents to offspring. And I want to know how many gametes this parent can make. Jeez Louise. I would never ask you to do this. A computer would be a sensible way to figure out the combinations of these letters. But you at least can determine how many there are, right? By using this formula, 2 to the n, where n is the number of heterozygous alleles. Let's count the heterozygous alleles in this crazy parent here. Is this heterozygous? No, that's homozygous dominant. But that one is, there's one. I guess I was trying to make that one heterozygous. Looks like a big C and a little c. I'll put a line there to make it look different. There's another one. There's another one. No. Yes, yes. 1, 2, 3, 4, 5, 6. So 2 to the 6th power equals, geez, I don't know. Plug it into a calculator. All right, we can do it. Let's see. 2 times 2 is 4, 8, 16, 32, 128. I think it's 256 gametes that this parent could make. And so you can see, you know, it can be a useful calculation, especially on this scale where you just want to know how many are there altogether. Okay. All right. So now what? I get really annoyed with students because they get this far and then they're like, oh, I don't know what to do. Well, you're pretty much done at this point, y'all. You just have to draw the box. Draw the dang box. Excuse my southern drawl. Okay. Draw the box, right? Well, now we have a great big box because we have, look what we have. We have, here, let me do it like this. This will make more sense. It'll save me some room. What if I do the box like this? I'm going to use these 4 as the top 4 of the box. Then I'm going to go down the side like this. So remember, the way the box works is you have the shape of the box is determined by the number of gametes that you have for each parent. Well, this parent has 4, so here's the 4 for this parent, and I'm just using those actual numbers there, or those letters. And then this one also has 4, so I put 4 across the top, 4 down the side. Do yourself a favor, do them in the same order if they're the same, just for bookkeeping purposes. Okay, now I got to try to make these horizontal lines. It's not the easiest thing in the world to do on this screen, but look at that. Okay, we got 16 boxes to fill in now. Remember about restoring homology. How do you fill this box in? Well, let's look at this one. You put the F's back together, you bring this over and this down, so you get big F, big F, big R, big R. Okay, there's that box. What about this one? Big F, big F, big F, little r. Now, stop looking at my boxes and you look at your own, you fill your own in. You should be drawing this with me. Here we go, big F, little r, little r. And I would advise you, take your time so that your penmanship doesn't screw you over, and it's easy for that to happen. Be careful. These letters look different, big case to lowercase, but be careful with, oops, see, now I made a mistake there. That's a big R. Be careful with letters that look the same, like C's, like you saw before. I couldn't tell if it was a big one or a little one. Do something to make one look different from the other, and you'll be glad. So we'll keep going here. We're getting close. Big F, little f, big R, little r. Big F, little f, little r, little r, little f, little f, big R, little r. And then this last homozygous recessive one, right, down here in the corner. All right. Now, what are we looking at here? This box represents all the different combinations of genotypes, right? And you could go through and figure out how many different ones there were and make a ratio. As long as you do the box correctly, then I'm just going to say that's good enough for the genotypes. But what about the phenotypic ratio? What's the ratio of the way the squirrels are going to look? And before we do that, we might say, well, irregardless of what we got with respect to these two traits, how might the squirrels look in general? Well, certainly you're going to expect some fluffy red ones, because those are both dominant traits, right? You could expect probably some fluffy albinos. You could expect some scraggly red ones. And you could expect maybe some scraggly albinos. Now look at these and tell yourself, these are literally the only 4 ways that the squirrel can look. It's either going to be fluffy red, fluffy albino, scraggly red, or scraggly albino. Now you see why you don't need to switch the letters around, right? Because think about it, a fluffy red squirrel and a red fluffy squirrel are the same thing, right? So don't switch the letters around. Now all you have to do to figure out this ratio is go through each box and figure out how many are fluffy and red, how many are fluffy albino, how many are scraggly red, how many are scraggly albino. So fluffy and red are both dominant. And what does that mean? It has to have at least one big F, could have 2, has to have at least one big R, could have 2. So let's go through and count all the fluffy red ones. What about this one? Fluffy red, it's homozygous dominant for both. Fluffy, it's heterozygous, but red is dominant. That one counts. Fluffy red, they seem all to be fluffy red. Fluffy red, fluffy red, fluffy albino. What about this one? Fluffy red, albino. This one is fluffy and red. This one is fluffy and red. Scraggly, scraggly. But here's one more, fluffy and red, albino, scraggly, homozygous recessive mutant squirrel, okay? How many do we get? 1, 2, 3, 4, 5, 6, 7, 8, 9 fluffy red in our ratio. Look at each box before we move on and tell yourself, I understand why every one of these is fluffy and red. Okay? Okay. What about fluffy and albino? If it's fluffy, it's still got to have one big F, could have 2, but if it's albino now, it's got to have what? 2 little r's, because that's a homozygous recessive trait. So let's find those. There's a fluffy albino. There's another fluffy albino. That one's red, that one's red. There's the third fluffy albino. Scraggly, scraggly. So there's 3 of those in the ratio. 1, 2, 3. Now we have 4 boxes left. Scraggly and red. Red is dominant, scraggly is recessive. So if it's scraggly, it's got to have what? 2 little f's, right? If it's red, it's got to have one big R, could have 2, and there's 3 of those. There's 1, 2, 3. Okay. And then this last box, we have this mutant squirrel here that's represented that is homozygous recessive for both traits. So you get one of those. This is a ratio, right? This is an expected ratio that you would get from breeding squirrels with these two traits. Okay? So this is awesome. I'm loving this. And we're going to do a practice problem now. I'm going to put the practice problem up there. I'm going to get you to work on it. I'm going to say pause the video, and then we'll do it together. Okay. So now here's the thing. You might be tempted to think, I could just memorize the ratios and then I don't even have to work the problem, but don't need to memorize the ratios. You need to work the problem because you don't know what I'm going to ask you to cross together, right? You have no idea what I'm going to ask you to cross together. So what that means is you need to know how to work the problem. If you know how to work the problem, you can always find the ratio. So here's a practice problem. It says in apples, red skin is dominant over yellow and tart is dominant over sweet. And then now it's telling you cross a homozygous dominant apple with a heterozygous apple, determine the genotypic and phenotypic ratios of the F1. You can only do the first cross in this example. You can't do the F2. So pause the video and work this problem, and then I'll be back to work it with you after you're done pausing. Okay, welcome back. Here we go. So always, 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 you assign alleles. Red is dominant over yellow. Big R. Oh, wow. Well, that's interesting. I didn't know. It just automatically it just automatically annotates in red, apparently. I didn't even push my little annotate button, and I have a little red dot. Okay. Red skin, dominant over yellow, so big R and little r. Tart is dominant over sweet, so we'll use a big, oh, I see the problem. The big T. Okay. I gotta do it different. Sorry. I see the problem now. Oh my goodness, it doesn't clear it. It doesn't clear it. Wow, that's fascinating. Okay, well, it is what it is. I'm learning new things about the program as we speak. So this is a big T, forget the red line there. There's a big R and a little r and a big T and a little t. Okay, and this is a mess, I know. Okay. Now it says, cross a homozygous dominant apple tree with a heterozygous apple tree. So if it's homozygous dominant for both traits, we're going to have big R, and big T, crossed with heterozygous. Hetero means one of each, so a big R and a little r, and a big T and a little t. So we're crossing these two parents together. Now this first parent, when we go to figure out the gametes, remember the R's and the T's get pulled apart. There's no options here really, right? You're going to get a big R and a big T. It's the only gamete this parent can make. Here though, we have some options. We could get a big R and a big T. We could get a big R and a little t. We could get a little r and a big T. Or we could get a little r and a little t. Now, I like this problem. And I like it because it shows you, it demonstrates that your box doesn't have to be square. We have one gamete from one parent and 4 from another. Okay, so check out what our box looks like. Looks like this. We got our big T, we got our, oops, little t, little r, big T, little r, little t. Sorry, I'm not writing very well. I'm doing my best. Okay, so we get this rectangle because we had one gamete here and 4 here. Fill it in, big R, big T, big R, big T, little t, big R, big T, big R, big T, little t. Okay, check these out. Here's our 4 genotypes. How many different genotypes do you see? And what's the ratio? What's the genotypic ratio here? Well, are any of them the same? No. The genotypic ratio in this problem is 1 to 1 to 1 to 1, right? These are all different. That's homozygous dominant. That one's homozygous dominant for one and hetero for the other. This one is sort of the opposite. It's hetero for that trait and homozygous dominant, and then this one is hetero for both traits. So there's 4 different genotypes, but what about phenotypes? How many different phenotypes are there? And what are they? What about this one? It's red and tart. Tart is dominant. Red and tart. Red and tart. Red and tart. They're all the same phenotype, right? There's no genetic variability among the phenotype of these 4 individuals, or these 4 genotypes. Okay, so here's a good lesson. Don't memorize the ratio. Learn how to work the problem, and then you can always figure out the ratio. Okay, that's my advice. All right, team. We're off to the next slide. I wish I could understand why this red won't go away. Can I erase it? Apparently not. Maybe the red is going to be on my PowerPoint forever. I don't know. Jeez. Okay. Move on to the next problem. We may not get to the 4th problem. I'll probably do a second video because we do still also need to do mutations. And so we'll see. We'll see how it goes. See how far we get. We're about 30 minutes in at this point. Okay, wow, look at all this. So now, here's the thing. So far, what we have seen is that all the traits that we looked at only had 2 alleles, right? A big R and a little r, for example, or a big F and a little f in the case of the squirrels. But it turns out that you can have multiple alleles. In other words, there can be more than 2 alleles for a trait. Human blood type is probably the greatest example of that I can think of, okay, where there are 3 alleles. And I'm just going to tell you the I stands for immunoglobulin. Immunoglobulins are proteins, and they're complements of proteins. And so it turns out there are 2 dominant alleles. We use the I to represent immunoglobulin, and then we're designating one dominant allele as being I to the A, right here. That's one allele. We have a second dominant that's I to the B. And then we use a little I to represent recessive. Okay, so these 2 are co-dominant alleles. Now here's the thing, as an individual, and then this one is recessive, okay? As an individual, you only get 2 of the 3. Everyone that's listening to this video has 2 of these 3 alleles. Could be 2 of the same one, it could be 2 different ones, okay? So how does this correspond to the blood type? And just to be clear, this is not the RH factor, plus and minus. This is not the part of the blood that we're talking about. We're just talking about your basic blood type, regardless of if it's positive or negative. That's a separate set of proteins. Okay, so here's the deal. Here, what are the possible combinations of these letters? Well, an individual could get two I to the A's, or they could get an I to the A and a little I. Either way, we would say they had type A blood. So this individual would be type A homozygous dominant. this individual would be type A heterozygous, right? For type B, same thing, except with Bs. Okay, so these have the B immunoglobulin proteins. Okay, but what if you get one of each of these, an I to the A and an I to the B? Well, they're both dominant. So we can't say you get I to the A or I to the B, so what do we call it? We call it type AB. We give both of them equal weight there, okay? And then the last possibility is homozygous recessive if you got two little i's, and we call that type O. Now, some interesting things, you may know this. Type O is the universal donor for blood. And it's the universal donor because it lacks those proteins on the surface. And what we know about the proteins on the surface is this is what causes your immune system to react, right? We said these act as antigens and your immune system doesn't recognize them, and they will attack them, basically, create an immune response. And so because type O individuals don't have these proteins, their blood can go into any of these other people and there's no proteins for their bodies to react to. Okay, so you can't put type B blood in a type A person or type A blood in a type B person because they wouldn't recognize the proteins and then they would attack the blood. And this would be a big deal. So what about type AB? Well, these individuals are lucky because they're the universal receiver. They can take blood from anywhere. Okay? So that's sort of explains blood type in us. And, you know, how can we turn this into some sort of a genetics problem? Well, pretty easily, as you could imagine. It plays out as a monohybrid cross because we're going to just look at one trait. And so let's look at an example of how this is going to work. You might also think because these are dominant that they're the most common, but they're actually not the most common. Type O in the United States ends up being the most common blood type. The way we explain this is because we believe that these immunoglobulins, the I to the A and the I to the B, are mutations which occurred relatively recently in human evolution, and they simply haven't had a chance to spread throughout our human population. There are some countries like Germany where type A is finally taking over type O. We would expect eventually type A and type B to be the dominant blood types because they are dominant alleles. Now, one of the things that we've learned about COVID, at least at the time of this recording being made, is that your blood type may have an effect on the severity of it in some individuals, and it turns out that type O seems to be less reactive to COVID and cause less problems than these others. Okay, so here's a good example. Okay, let's say Mission Hospital, 3 o'clock in the morning, 2 mothers in there giving birth, and it's late. The nurses are exhausted, and what happens is they snatch the little babies from the mamas and they put them in the little baby nursery, but they forget to put bands on their wrists. And so now we have these 2 babies, right? And unfortunately, we don't know, the nurses can't remember which babies go with which parents. Okay. So we have baby Jane Doe number 1 and baby Jane Doe number 2, and then we have the Rap Chicks and the Hughes. And, you know, the mamas are getting a little antsy. They're like, you know, can we see our kids? And the nurses are like, ooh, eeny, meeny, miny, mo. Like, what are we going to do here? Okay. Can a simple blood test determine what baby goes with what parents? That's the question. And so given what I've told you about blood types, you should be able to decipher this. The way to approach this is to turn the blood type into genotype or genotypes, okay? So for example, if baby Jane Doe number 1 has type A blood, do we know for certainty what baby Jane Doe's genotype is? Well, not yet, but we could say it could be that baby Jane Doe is homozygous dominant type A, or baby Jane Doe could be heterozygous type A. Either one is a possibility. Okay. What about baby Jane Doe number 2? Baby Jane Doe number 2 has type O blood. Well, we do know with certainty her genotype, because the only way you get type O blood is if you are homozygous recessive. So she has two little eyes. Mr. Rapchick, same deal. He has 2 little eyes. Mrs. Rapchick, though, I to the A, I to the B, we do know with certainty her genotype. And then the Hughes, well, we don't know for sure. We know Mr. Hughes could be homozygous dominant or heterozygous type A. This is a semicolon here, sorry. It's confusing. Let me rid myself of the semicolon so you don't think it's a little I in this case. Here we'll just do a comma, how's that? And then Mr. Hughes or Mrs. Hughes, same thing, but with type B. So can we do it? Can we determine which baby goes with which set of parents? And, you know, you might look at this and see 2 little eyes here and 2 little eyes here and say, oh yeah, baby Jane Doe 2 goes with the Rapchicks. That would be an incorrect assumption. Can you think of why? Although Mr. Rapchick has 2 little eyes, he only donates one of them to the baby, right? And Mrs. Rapchick donates the other allele. So it turns out that if you cross a type O and a type AB, let's do it. If we cross an I to the A, I to the B individual with a little I, well, the type O only can donate a little I. Here we can get an I to the A or an I to the B, right? So when you fuse back together, you see, okay, only blood types these parents can make are I to the A little I or I to the B little I. Interestingly enough, a type O and a type AB can't make either of those blood types back out. They can only make a type A heterozygous or a type B heterozygous because you only get one allele from each parent. Okay, so that's important to understand. So then what that means is the type O baby here, let me get rid of this to make room. What that means is the type AB baby goes down here with the Hughes. Now, with that said, now do we know what their genotypes are? We certainly do. If they're making a type O baby, then what that means is they both got to have a little I. And so we can eliminate this genotype and we can eliminate this genotype and we can say safely Mr. Hughes is heterozygous type A and Mrs. Hughes is heterozygous type B. That then means that baby Jane Doe number 1 goes over here with the Rapchicks. And now look what we can do. We can use the parental genotypes to eliminate one of these genotypes, right? So we know that what happened was the baby had to get a little I from Mr. Rapchick. And so that means that we can eliminate this homozygous dominant genotype. And so it just turned out that the baby got a little I from his dad or her dad, and an I to the A from her mom. Could have just as likely gotten a I to the B, but didn't. Okay, so there you go. So these are the ways that you can do these problems. You can work backwards. I can give you genotypes of offspring and ask you to tell me something about the parents, or we can work forward. Okay, and work in either direction. So here's the thing, there's one more type of problem, and I'm going to introduce that type of problem just very briefly, and then we will save that for the next video, because we're also going to do mutations, and I don't think I can want to cram all that into what time we have left, okay? So we've done well. We've made it through most of the problems, most of the types of problems. And I would encourage you to start working and looking at those sheets, because you can work any of the problems except for the ones dealing with polygenetic inheritance, which we won't get to in this video. We'll do it on the next video. Okay, so what we're looking at now in the last unit is called polygenetic or multiple factor inheritance. And in this case, you have more than a pair of chromosomes controlling the trait. And a good example of that is human eye color, where it's like this: 1, 2, 3, 4, 5, 6, 7, 8 different chromosomes, 4 homologous pairs, all have a gene for eye color. So they each literally are contributing an eighth to the color of your eyes, right? Combine that with multiple alleles, and man, it can get complicated. And that's why if you looked at a room of people, you would see every imaginable shade of eye color, from the darkest dark to the palest gray, right? Okay, so we'll come back to this next time. What we have left in this unit for genetics, at least, is to finish up this type of problem and then talk about genetic mutations, which is a fascinating topic. And then we move into protein synthesis. And then after protein synthesis, we'll be ready for our next unit test. Okay, team. I will see y'all next time. Bye-bye. Have a good day. Enjoy yourselves. Don't forget to enjoy yourselves. It's important in life. You have to enjoy yourself.